let's talk about the epigenetic regulation, get back to the story about, how long ago was that, a week or so? We were talking about sexuality or sexual orientation in epigenetics. So again, the outcomes for understanding, for finishing epigenetics, that's what we do today. Then we come back a week from now and start talking about how eukaryotes, like humans, regulate gene expression, briefly. So appreciate how our environment impacts our gene expression, and then also I'd like, by the end of the class, one of the assignments is for you to create a gene regulatory network. Imagine, create, be creative. Gene regulatory network, though. The last time we were together, a bunch of you completed this kindergartenish assignment to color in the cat based on whether or not it's advanced kindergarten. Color in the cat whether or not you think X chromosome inactivation was random or, what was the other word? Skewed, that's right. Non-random. So there's random, these are 3 different submissions from 3 different students, and skewed on the bottom. And we talked about this last class briefly. So these 3 I just picked to illustrate the point that the random X chromosome inactivation, when you have one X chromosome that has an orange coat color gene and the other version has a black coat color gene, in the random sets, about half of the cat is orange and about half the cat's black, meaning about half the cells in the cats on the top randomly turned off their orange pigmentation gene, and the other half of the cells in those cats randomly turned off their black pigmentation gene. And the skewed cats, it's not half and half. It's not 50% orange, 50% black. It varies on which cat you're looking at, but it's either 95% orange or what, like 75 or 80% black. So that's what the definition between random X chromosome inactivation and skewed X chromosome inactivation means. So thanks for helping me define that. Now we're going to use this information. Do you have any questions before we move on? X chromosome inactivation. They get randomly turned off. You expect half of the cells or half of an organism to have one X chromosome expressed and half the other. The calico cats on the top, when you look at their chromosomes, of course not their coat colors, or do the moms look more like the cats on the bottom, where there's an extreme bias towards which X chromosome the moms of the homosexual men turned on or turned off. And it turns out that some of these same authors that did the study of the twin study of human sexuality that we talked about a week or so ago also did this earlier study looking at X chromosome inactivations in the mothers of those sets of gay male twins. So what did they find? So you get a bunch of pairs of twins, homosexual or not. What are they asking about X chromosome inactivation? Extreme skewing of X chromosome inactivation. So if we were looking at the moms of the gay men, what are we asking? Do the moms look like? Here are control mothers. What's on the x-axis, for those of you that are up close to your tablets and can see the x-axis? What do we expect? It's a fraction of cells inactivating the same X chromosome. So say we're looking at the first X chromosome, X1, not X2. What do you expect for random X chromosome inactivation? What fraction of cells would turn off X1? Half. So we would expect to see 50% inactivating X1. So we'd expect to see, say, a column there. Right? If every single woman turned off exactly half of X1 in their cells, and the other half of their cells turned off the second X chromosome. So what does it mean that we have this type of histogram? Where's 50%? This column. So out of 103 moms, what is that, 6 on the y-axis? 6 out of 103 moms had exactly 50% random, what we would call random X chromosome inactivation. What was the most common frequency of X chromosome inactivation in these moms for this plot? What is it? It's about, 70 to 75%. So 20 out of 103, about a fifth of the mothers, almost 3/4 of their cells randomly turned off one X chromosome, not the other. Okay. And then up here at the very top, where over 90% of the cells in that mother turned off one X chromosome, there's like 3 or 4 individuals. This is what they're defining, the black columns up here is what they're defining as skewed. Like the calico cats that are mostly orange or mostly black. Mothers of non-homosexual men generally have what we would consider random X chromosome inactivation. What happens when you start looking at the mothers of the gay men, though? How does this plot in the upper left differ from the one in the upper right? What's changed? Okay. Moms of heterosexual men on the left, mothers of gay men on the right. What's the main difference? If you took the histograms and laid them on top of each other, where would you see a big change? It's one of those black columns. Now we have, when you just switch from looking at mothers of heterosexual men to mothers of homosexual men, you see this huge increase in the number of mothers that have extreme bias towards turning off one X chromosome and not both either randomly. And this effect is mostly due to the mothers of more than one gay son. So that huge column at 90 to 95% of X chromosome 1, say, being inactivated, that comes mostly from those mothers that have multiple gay sons. Weird. So epigenetics, is this involved in sexual orientation? Like the story we talked about last time, can you predict who might be homosexual based on your mom's X chromosome inactivation pattern? What do you think? If I told you that somebody's mother had 90 to 95% inactivation of the same X chromosome throughout her entire body, would you place money on betting that her son is homosexual? It's only 10 out of 44, it's only a quarter of the moms that exhibit this variation. So it's actually a pretty bad bet, 10 out of 44 mothers having X chromosome inactivation patterns that are that skewed. So these authors said, well, there's some sort of an association, correlation, not necessarily causation, and move on. And that's how we got partly to the story that we talked about a week ago. They go on to look at other sorts of epigenetic changes in the twins. But there might be something about the maternal environment that the fetus develops in that controls sexual orientation. So your mom's epigenetic patterns might matter. Sure.
Okay, so we're saying that just looking at the right side,
Yep.
it kind of makes sense, but if we look at the left, they're all skewed left, and they still have homosexual sons. So how does that make sense in terms of the data?
So we're talking about, so you mean these are all greater than 50% X chromosome
Looking
inactivation?
at the graphs of the mothers of gay men and mothers of 2+ gay sons.
Yes.
On the right side, it makes sense.
Yeah.
On the left side, it's skewed to the left.
Right.
So how does that, in terms of data, make sense?
So that's interesting. So we have to look at.
They still had average, or not average, but standard 50%.
Yeah, 5. So one of the important things to do comparing these two graphs is to look at the scale of the y-axis, which might explain partly that of these 13 moms, 8 of them had 50% to 55% X chromosome inactivation when they had one gay son, and the other 5, so right, 5 plus 8 is those 13. So that graph is just split into those 2 data sets separately. And so it is interesting that these numbers, 5, 6, 7, and 9, are smaller than in that graph. So that suggests that the mothers of 2+ gay sons, they don't have the same distribution, no. But if these were scaled the same height as that graph was, these columns would be shorter. Does that make sense? Because really that graph breaks down into those 2 graphs. So we could keep the y-axis the same. I'm not sure if that detail really is important. Totally missed your question, but do my best to answer. Let's connect after class if we can break this down a little bit further. Please. Yeah, okay. Okay. So the other story about epigenetics, which I think hopefully will be even more interesting, hits close to home because now we're actually talking about people that live in Fresno. We're talking about air quality and we're talking about asthma. It's really horrible. So the American Lung Association puts out this report every year, the state of the air, and of course what they do is they trash the cities that have the highest air pollution levels, which of course, let's see, 25 counties most polluted by year-round particle pollution. On top, Madera County. Second, Kern County. Third, Kings County. Tulare County, Fresno County, Stanislaus County, Riverside County, Empirical County, San Joaquin County. So basically, we are surrounded by like the top 10 most air-polluted year-round counties in the nation, which you already knew, but here are the data. So year-round, we're being exposed to really high levels of air pollution. And recently I saw this kind of cool paper about air pollution and epigenetics and asthma in the Central Valley. So this is just the citation of the paper, but it's the first author here, Carrie Nadeau, is a pediatrician who works at Stanford or in Palo Alto. And so this is a story about trees versus the dogs. So we've got Stanford over here in Palo Alto, and we've got Fresno State. And this pediatrician who practices in Stanford started seeing a lot of kids coming to her clinic presenting with asthma. And at some point she started wondering, where are all these kids with asthma from? Because normally a lot of kids in Stanford and Palo Alto area don't have as much allergy as Central Valley. And she started looking and she discovered that of all of her patients, the ones that came from zip codes in the Central Valley were the ones that were coming to her complaining about asthma, and not kids from Palo Alto or pretty much anywhere else in the state. Big shock. That's why we have so many asthma doctors in Fresno, right? But she's a medical scientist. She's not just a physician, she's also a researcher, and she had this hypothesis that maybe what was happening, based on some other research that had been done previously, she thought there must be something happening to the immune system cells of kids that live in the Central Valley compared to kids that live in Palo Alto. So don't freak out. Here's immunology, not even 101, immunology 1. My understanding, because I'm not an immunologist. So what happens is, how you get asthma, there are these cells, antigen-presenting cells, that essentially carry the signal that, hey, there's something in the environment that I'm allergic to or responding to. Those cells pass that signal on to cells called Th0, or T helper cells, T helper. And those Th0 cells are not cells that have ultimately decided what sort of cell they're going to become when they grow up, kind of like me. They haven't differentiated yet. So those Th0 cells, depending on the type of signals they get, which are all of these letters and number combinations written along the arrows. So for example, if a Th0 cell is stimulated by the molecule TGF-beta, that would cause the Th0 cell to turn into a Th17 cell. The cells that we're particularly curious about here are the Th2 cells, which are involved in asthma and allergy. And what happens is if you get too many of these T helper 2 cells, that's bad. So these are the things that cause allergic reactions, and the mucous membranes start producing all the snot and all that sort of thing, gumming up your airway, making you have difficulties breathing. So it's, of course, as you probably all know, it's good and bad to have an immune system. It's good that your body tries to fight this off, but the effect that your immune system can have on the body when it's overstimulated is what causes asthma and a bunch of immune disorders. So we also have cells, the T regulatory cells, Tregs, that work to repress, that's what that arrow with the blunt line is. So T regulatory cells repress our T helper 2 cells. These are the things, the Tregs are the things that keep most of us from having allergic reactions and severe asthma when our body starts producing T helper 2 cells. We also produce some regulatory cells that stop those T helper cells from going crazy in our bodies. So her hypothesis about the kids that were presenting with asthma from Fresno was maybe they don't make enough T regulatory cells. So they respond to something in the environment, the kids do, their bodies do, and they produce a lot of T helper cells to try to fight off those invaders, chemicals, small molecules, whatever, air pollution. But if they don't have enough regulatory T cells, that causes asthma and other related symptoms. So then she had to look at some additional papers to figure out how is it that T regulatory cells get created in the first place. And aside from TGF-beta and IL-12, there's another gene, FOXP3, that's also involved in creating T regulatory cells from that initial population of Th0 cells. So if you want to make regulatory T cells, you have to have the FOXP3 gene turned on. We're talking about transcription now, so hopefully we're going to start connecting the dots. We're talking about asthma and the critical role of the FOXP3 gene in making regulatory cells. Any questions to this point? So this is her hypothesis. She's going to test this hypothesis by looking at the patients, collecting their immune system cells, and then asking about how functional on the y-axis their Treg cells are. So I'm going to ask you to help me interpret this graph. So put the key down there below. In red, I'll circle the individuals that have asthma, and in blue, we'll have NA, or no asthma. But what does this mean? If you're healthy, what do you want? Better functioning Treg cells or lower functioning Treg cells? These are the ones that prevent your immune system from going crazy when you're exposed to allergens and other stimulants. So you want them to be functioning better. So the goal of the body is to have your Tregs functioning. Which group has the best functioning T regulatory cells? Stanford kids that don't have asthma. So kids that don't live in the air pollution, that also don't have asthma, have really high functioning T regulatory cells. Okay, what group is next lowest? The Stanford kids that have asthma have better functioning T regulatory cells than the Fresno kids that don't have asthma, a little bit. And the Fresno kids that have asthma, horrible function of these T regulatory cells. So maybe it's not surprising that a lot of people in Fresno have asthma and related respiratory symptoms if our T regulatory cells aren't able to keep us from having our T helper cells go into overdrive when we're exposed to pollutants. So I'd like you to make a prediction now. So we've talked about the FOXP3 gene, its transcription, producing the FOXP3 protein is essential for making these regulatory cells. And we just saw that kids in Fresno that have asthma have really crappy T regulatory cells in general. Here's the connection. studies in other organisms have suggested that epigenetic changes can modify expression of the FOXP3 gene. What sort of epigenetic changes would we be talking about here?
Pollution.
Pardon?
Pollution.
So pollution might be the trigger,
Oh, methylation.
but then, yeah, so then somehow there's a connection potentially between exposure to toxins, air pollution, and the methylation of the FOXP3 gene. So we talked last 2 classes ago about how methylation of a promoter can prevent transcription by blocking transcription factors from binding to the promoter and turning on transcription. So usually methylation of promoters means you turn off gene expression. So here's the prediction I'd ask you to make. Don't say it out loud yet. Is FOXP3 more heavily methylated in Palo Alto kids or Fresno kids? And would it be more heavily methylated in kids with or without asthma? So on the next slide, I've got a blank graph here. So I'd like you to draw what you predict. This is all anonymous. I'd like you to draw what you predict this plot will look like when we look at FOXP3 methylation levels. Okay, so really quickly, let's set up the axes first, and then I'd like everybody to plot this. And take a screenshot, submit it to the Google exercise that I posted at the start of class today. Yes, sir.
Quick reminder, the other opposite to methylation is acetylation.
Right.
Does acetylation increase the transcription?
So we don't know for FOXP3 if acetylation increases transcription like methylation decreases it. But an excellent question, because that's a really critical thing for everybody to remember, that methylation and acetylation usually have opposite effects. Okay, so if we're measuring FOXP3 methylation, what's going to be on the y-axis? What label should we put there? So, yeah, what about FOXP3 do you want to put on there?
The protein.
Number of methyl groups in the promoter, or fraction of, pardon?
Percent methylation?
Okay, percent methylation. Okay. Of the FOXP3 promoter. And then what are we going to put on the y-axis, or the x-axis? For this prediction. So we're measuring this methylation pattern in what groups of individuals? Independent variables. So different kids. So we're going to use the same groups. So we'll do Palo Alto, what was it? Asthma. Palo Alto, no asthma. Fresno, asthma. Fresno, no asthma. So take a couple of minutes, draw what you think the data would look like. Just one data point per column. So what do you think the pattern is going to look like? And then take a picture of that and attach it to the Google Classroom assignment. Do feel free to work with a friend if you want.